Hello, my dear students. Welcome you all to Shri Jain Rank Zone classes. I am a mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the previous videos of human respiratory system, which is a part of human physiology, we discussed about different air capacities, lung capacities, air volumes or pulmonary volumes. We discussed about the anatomical features of lung. We discussed about the respiratory tract. And today, after the discussion of exchange of gases in the alveoli on the concept of partial pressure and the rate of diffusion, we are going to start with transport of oxygen. As you know, gases diffuse from high pressure to low pressure. The pressure of the gas is basically a very important factor in diffusing through the biological membranes or the alveolar epithelium. Oxygen is transported mainly through hemoglobin. Oxygen is carried by blood in two forms, in solution and as oxyhemoglobin. It is transported in the form of solution. In the form of solution means when oxygen combines with the plasma, and oxyhemoglobin when oxygen combines with hemoglobin, Hb. RBC, red blood corpuscles, they have hemoglobin, and as the shape of RBC is biconcave, it provides large surface area for oxygen transport. As you know, mature RBCs are enucleated. They don't have nucleus because having nucleus will become a problem in transporting maximum amount of O2 because that nucleus only will occupy the whole space. And that is why the mature RBC is enucleated. In solution, in the form of solution, oxygen is soluble in plasma to very small extent under normal conditions of temperature and pressure. When we talk about the oxygen gas is soluble in plasma to very small extent under normal conditions of temperature and pressure. Hence, most of it is carried by red blood corpuscles (RBCs) in the form of oxyhemoglobin. About 3% of oxygen is transported by blood in dissolved form in plasma of blood. Around 3% of oxygen is transported by blood in dissolved form. Rest all of the oxygen, in around 97%, is basically transported by the RBCs or red blood corpuscles, right? Now, coming to next, out of 4.6 ml of oxygen entering each 100 ml of blood in lungs, only 0.17 ml travels in the solution form in plasma. Out of 4.6 ml of oxygen entering each 100 ml of blood in lungs, only 0.17 ml travels in solution in the form of plasma, or it combines with plasma, but it only transports 0.17 ml in the form of soluble oxygen. Okay, because oxygen gas is less soluble, we can say it is very less soluble in the form of plasma. Now, coming to next, as oxyhemoglobin. RBC contain a protein called hemoglobin. RBC red blood corpuscles, they mainly contain a protein hemoglobin in which heme is pigment. Heme is red colored iron based pigment. So basically hemoglobin is a metalloprotein. It has 4 polypeptide chains and 4 heme groups. It is basically having 4 polypeptide chains. When we talk about adult hemoglobin, 2 alpha chains and 2 beta chains, and it is having 4 heme groups attached to it, or 4 atoms of iron in ferrous form, Fe2+. Thus, it can react with 4 molecules of oxygen to form Hb4O8. Okay, so 4 molecules of oxygen can combine with one hemoglobin, because in one hemoglobin molecule there are 4 heme groups having 4 atoms of iron. Thus, it can combine with 4 molecules of oxygen to form Hb4O8 or HbO2. In simplified form, you can also write HbO2. This is called oxyhemoglobin. Iron in ferrous state is converted to ferric state after getting oxidized. Fe2 plus get oxidized to Fe plus 3. Okay. This combination is called oxygenation. When combination or the combining of oxygen with hemoglobin is known as what? Oxygenation. On an average, 15 gram of hemoglobin is present in 100 ml of blood. In adult person, around 15 gram of hemoglobin is present in 100 ml of blood. 1 gram of hemoglobin combines with 1.3 ml of O2. Very important. Around 15 gram percent of hemoglobin concentration in adult person. 1 gram of hemoglobin combines with 1.34 ml of O2. Thus, 100 ml of blood carries approximately 20 ml of O2, 19.4 ml to be exact. Okay. So 15 gram of hemoglobin is present in 100 ml of blood. 1 gram of hemoglobin combines with 1.34 ml of O2. Thus, 100 ml of blood carries approximately 20 ml of O2, 19.4 ml to be exact. But when blood reaches the tissues, the oxygen concentration reduces because the dissociation of oxyhemoglobin takes place. Dissociation of oxyhemoglobin takes place gradually to 14.4 ml, which is collected by venules and vein. So when blood reaches the tissue, the oxygen concentration reduces gradually to 14.4 ml because of the collection of oxygen by venules and vein. Thus 5 ml of O2 is transported by 100 ml of blood under normal condition. That is why the 5 ml of O2 is transported by 100 ml of blood under normal condition. So out of 19.4 ml or 20 ml, 14.4 ml is collected by venules and vein, and only 5 ml of O2 is left behind for the transportation by 100 ml of blood under normal condition. Hemoglobin has higher affinity for oxygen, and this affinity is increased by fall in partial pressure of CO2 of blood. Partial pressure of CO2 gas in blood. Hemoglobin has higher affinity for oxygen, and this affinity increases by decreasing the partial pressure of CO2 gas in blood. Okay, so what are the facts you have to remember from this slide? That RBC contain a protein which is known as what? Hemoglobin. Heme is a metalloprotein having a red colored heme pigment, okay, in which iron is present. There are 4 atoms of iron which are present in the ferrous form. It contains 4 heme groups. Each iron is placed centrally in between the porphyrin ring, and that porphyrin ring is basically having 4 pyrrole rings, sorry. Thus it can react with 4 molecules of oxygen to form Hb4O8 or HbO2. This is called oxygenation, and the formation of the product is oxyhemoglobin. 15 gram of hemoglobin is present in 100 ml of blood. 1 gram of hemoglobin combines with around 1.34 ml of O2. So 100 ml of blood carries around 19.4 ml exactly, or 20 ml approx. Out of this 20 ml, 14.4 ml is collected by venules and vein, and only 5 ml of O2 is actually transported by 100 ml of blood in normal conditions. Hemoglobin has higher affinity for oxygen, and this affinity is increased by decreasing the partial pressure of CO2 gas in blood. Structure of oxyhemoglobin. There are 2 forms. One is the relaxed form, and the other one is taut form. The membrane of RBC It's a classical example of Donan's membrane or Donan's equilibrium. This Donan's membrane, when combined with oxygen, okay, the oxygen make out some changes and the relaxed form, okay, is created. This three-dimensional structure gets changed, the conformation gets changed to taut form or tense form when oxygen is less. So at low oxygen concentration, taut form is there because of the presence of strong hydrophobic bonds, okay, and weak ionic and hydrogen bonds. Taut form is there or tense form, okay, and the oxygen disrupts the weak ionic and hydrogen bonds, okay, which causes the relaxed form or high oxygen affinity. One O2 binds a hemoglobin subunit. O2 binding disrupts interdimer bonds, okay, and increasing O2 affinity remaining subunits increases. So this is known as what cooperative binding, the conversion of relaxed form into the taut form when oxygen is taken up by hemoglobin. Each subunit resembles myoglobin tertiary structure. Each subunit of hemoglobin resembles to the in this subunit like beta form. This is beta form, this is alpha form. So each subunit resembles a myoglobin tertiary structure. Next slide. At the alveoli, venous blood At the alveoli, venous blood has low oxygen and is exposed to low partial pressure of CO2 of alveolus. Thus, oxygen diffuses into red blood corpuscles and form oxyhemoglobin, bright red in color. At the alveoli, venules blood or the venous blood has low oxygen tension, has low oxygen concentration, and is exposed to low partial pressure of CO2 in alveolus. Thus, oxygen diffuses into red blood cells and form oxyhemoglobin, bright red in color. As CO2 diffuses from the blood to alveolus, blood partial pressure of CO2 falls, increasing further uptake of oxygen. Okay, so it is like when alveolar blood it exchange or the exchange of gases takes place at alveoli, the partial pressures at the alveoli is around mean the alveolar wall is one cell thick, because the thickness of alveolar wall will decide the rate of diffusion. Okay, so at the level of alveoli, when we talk about the level of alveoli, the partial pressure of oxygen is around 104 millimeters of mercury and partial pressure of CO2 is around 40 millimeters of mercury. Okay, but the blood which is carrying the oxygen is very less, mean around 40 40 millimeters of mercury, and the blood pulmonary artery blood carrying the CO2 partial pressure of CO2 is around 45 millimeters of mercury. So at the level of alveoli, this is alveoli and this is pulmonary artery, sorry. So alveolar air basically having partial pressure of O2 104 millimeters of mercury and partial pressure of CO2 40 millimeters of mercury. And pulmonary artery carries the partial pressure of oxygen 40 millimeters of mercury and PCO2 46 millimeters of mercury. The diffusion of CO2 gas is much faster in comparison to oxygen gas. Okay, although the gap is very less, but the CO2 gas diffusion rate of diffusion is much faster. Okay, it is twice in comparison to oxygen gas. Okay, so oxyhemoglobin remains unchanged. It remains unchanged till it reaches the tissue where it dissociates readily to release oxygen. Because at the level of tissues, when the pulmonary vein comes out of the, it carries the blood to the left atria and left ventricle. The pulmonary vein is having partial pressure of oxygen 95, while partial pressure of oxygen is 40. Okay, so when it reaches to systemic arteries, the partial pressure remains same. There is no change. Okay, but when it comes down at the level of capillaries, at the level of capillaries, The all of the oxygen is basically given out to the venules and veins. Okay, only 5 ml per 100 ml of oxygen is transported. Okay, so oxyhemoglobin remains unchanged till it reaches the tissue where it dissociates readily to release oxygen, because at the level of tissues partial pressure of CO2 is very high in comparison to partial pressure of O2. Okay, is that understood? Now, structure of porphyrin. This is a very important topic, structure of porphyrin. Porphyrins are the cyclic molecule, and these porphyrins, they are basically made up of four pyrrole rings which are linked by methylene linkages. Okay, methanol bridges or methylene linkages. There are 8 side chains, two on each pyrrole. Okay, two on each pyrrole. These are the methylene linkages. These are they can be the vinylic linkages. Okay, these side chains are acetyl, propionyl, methyl, and vinyl. Different porphyrins vary in structure of different side chains depending on the substituent groups, dividing into first series systemic arrangement, third series asymmetrical arrangement. First series arrangement of porphyrin is symmetrical, third series arrangement of porphyrin is asymmetrical, although don't have to know, just have to remember this. Fe is present in the +2 form. Okay. So this is the structure of porphyrin, which basically contain four pyrrole rings. These are the four pyrrole rings. Okay. These pyrrole rings are also having the phytol chains. Okay. Now coming to next, oxyhemoglobin dissociation curve. Very, very, very important. The percentage of hemoglobin that is bound with oxygen is called percentage saturation of hemoglobin. Very important. The percentage of hemoglobin which is basically taking up the oxygen is called percentage saturation of hemoglobin. The relationship between the partial pressure of oxygen and percentage saturation of hemoglobin with oxygen is graphically illustrated by a curve called oxyhemoglobin dissociation curve. Under normal conditions, the oxygen hemoglobin dissociation curve is sigmoid shaped or S shaped. The lower part of the curve indicates the dissociation of the oxygen from hemoglobin. The lower part of the curve basically indicates the dissociation of oxygen from hemoglobin. Under normal conditions, the oxygen hemoglobin dissociation curve is sigmoid shaped or S shaped. The lower part of the curve indicates dissociation of oxygen from hemoglobin. The upper part of the curve indicates the acceptance of oxygen by hemoglobin. When the partial pressure of oxygen is around 55 millimeters of mercury, the hemoglobin gets saturated to around 50%. It means the blood contains 50% of oxygen. The partial pressure at which hemoglobin saturation is 50% is called P50. At 40 millimeters of mercury, partial pressure of oxygen, the saturation is 75%. It becomes 95% when the partial pressure of oxygen is 100 millimeters of mercury. So please rectify this. This is not 255. This is 55. Okay. So the upper part of the curve indicates the acceptance of oxygen. Lower part of the curve indicates the dissociation of oxygen from hemoglobin. When the partial pressure of oxygen is 55 millimeters of mercury, the hemoglobin gets saturated to 50%. And 50% of hemoglobin is now saturated to oxygen. It means blood contains 50% of oxygen. The partial pressure at which the hemoglobin saturation is 50% is called P50. At 40 millimeters of mercury, a partial pressure of oxygen, the saturation is 75%. It becomes 95% when the partial pressure of oxygen is 100 millimeters of mercury. Hemoglobin does not take up the oxygen. Okay, hemoglobin does not take up oxygen in the low as it is having low partial pressure of oxygen, but as oxygenation of pigment occurs, its affinity for more O2 increases. Okay, in hemoglobin, where four subunits are present, acquisition of one molecule of oxygen increases the affinity of neighboring hemes for oxygen. This is known as cooperative binding. Okay, between active sites. I mean, it is like that when one molecule of oxygen combines with one heme, then it basically changes the conformation and it becomes relaxed. Okay, as the conformation changes, the other heme groups also increases the affinity for oxygen. Okay, this is known as cooperative binding between active sites. This is oxygen hemoglobin dissociation curve. You can see decreased P50 or increased affinity. Okay, temperature, PCO2, 2,3 dpg, okay, and pH values. These are the factors which basically control the dissociation and the association of oxygen to hemoglobin. Okay, oxygen to hemoglobin is basically controlled by all these, factors. Just a minute. Yeah. Hmm. So you can see the lower part of the curve. This part of the curve basically it resembles the dissociation of we can say the dissociation of this part of the curve represents dissociation of oxyhemoglobin, and this part resembles the upper part resembles the upper part resembles association of oxyhemoglobin. So the shifting of curve to right side or to left side. Okay, the shifting of curve to this is right side and this is left side. The right side shifting of the curve is seen when increased P50, matlab when P50 is increased, decreased affinity, okay, temperature is high, high partial pressure of CO2, high 2,3 dpg amounts, and when pH is less. Because when CO2 is more, then there is more formation of carbonic acid, H2CO3, which will make the blood acidic and decreases the pH. Okay. The shifting of left side of the oxyhemoglobin dissociation curve is basically because of the conditions like decreased P50, increased affinity, okay, temperature. Because if there is decreased P50, I mean there is more amount of or more affinity towards oxygen, okay. So when temperature is less, when partial pressure of CO2 is less, when 2,3 diphosphoglycerate levels is less, and when pH is high. Okay, 2,3 dpg is 2,3 diphosphoglycerate or diphosphoglyceric acid. Okay, so when this there is acidosis, okay, when decreased pH, then it will shift to the right hand side, and when the condition is alkinosis, okay, when pH is high, alkinosis, alkinosis, high pH, shifting to left side in more affinity towards oxygen. Acidosis, low pH, shifting to right side. Okay. Factors affecting oxygen dissociation curve. First is the temperature. At higher temperature, hemoglobin gives up oxygen more readily. Okay, the bond will break, and the dissociation curve shifts to right side. Now, please look. As the temperature is high, okay, the curve is shifting to the right hand side. That is of physiological importance because increased temperature means higher metabolic rate or higher oxygen requirement. Okay, if the temperature is high, tissues they need more amount of oxygen for or we can say if the temperature is high, it means the higher metabolic rate mean tissues requirement of oxygen is high. Increase in CO2 or other acids lower the pH of plasma and shift the dissociation curve to the right side. At higher CO2 concentration, more O2 is given up at any oxygen pressure. This is known as what Haldane effect. Okay. At higher CO2 concentration, more O2 is given up at any oxygen pressure. If the CO2 concentration is high, then beat whatever the pressure of O2 is, the oxygen will be released from oxyhemoglobin. Okay. Partial pressure of CO2. CO2 lowers the oxygen affinity of hemoglobin even if the pH is kept constant, even if we keep the pH constant, but if the CO2 levels are high, the oxygen hemoglobin dissociation curve will shift to right side, mean it will more dissociate. Okay, shifting to left side, it will more associate, mean shifting to right side, more decrease affinity towards oxygen. Shifting to left side, increase affinity towards oxygen. Oxygen dissociation curve shifts to right and to release more O2 with increase in partial pressure of CO2. 2,3 diphosphoglyceric acid or diphosphoglycerate. It is present in the red blood cells of adult, formed from 3-phosphoglyceric acid. It is formed from 3-phosphoglyceric acid. It competes for oxygen binding sites in hemoglobin molecule. As it binds to the beta chain of adult hemoglobin, it causes a right shift of the dissociation curve, resulting in higher P50. Okay. 2,3 diphosphoglyceric acid. It is present in the red blood cells of adult, formed from 3-phosphoglyceric acid. It competes for oxygen binding sites of hemoglobin molecule. As it binds to the beta chain of HbA, the curve shifts to right side, resulting in higher values of P50. Resulting in higher values of P50. Increased P50. Okay, means decrease affinity towards oxygen. Mean now the hemoglobin require more amount of oxygen or high pressure of oxygen to combine with it. Okay, although it is a very important fact that whatever be the pressure, partial pressure of oxygen, whether you increase it, okay, but if the CO2 levels are high, then it will dissociate oxygen. Okay, it will not combine with oxygen. Lower CO2 concentration, lower body temperature, lower 2,3 dpg, lower the P50, and the curve moves to left side. Okay. If all things are, I mean, you can say, if all things are lower, decrease P50, decrease temperature, decrease PCO2, decrease 2,3 dpg, it will shift to left side. Okay. So shifting to right side and shifting to left side, I hope everything is understood. Okay. Bohr's effect. Shifting of oxygen hemoglobin dissociation curve to the right by increasing carbon dioxide partial pressure is known as Bohr effect. If we increase the partial pressure of CO2 gas, then the oxygen hemoglobin dissociation curve will shift to the right side. Okay, it will shift to the right side. This is known as what? Bohr's effect. It is known as what? Bohr's effect. It is named after Danish physiologist Christian Bohr. Okay. So what is Bohr's effect? Shifting of the oxygen hemoglobin dissociation curve to the right by increasing CO2 levels, CO2 partial pressure is known as Bohr's effect. The presence of CO2 decreases the affinity of hemoglobin. The presence of CO2 gas decreases the affinity of hemoglobin for oxygen and increases the release of oxygen to the tissues. If more amount of CO2 is there, high pressure of CO2 is there, then the affinity of hemoglobin towards oxygen decreases and it will release more and more amount of oxygen to the tissues. The pH of blood falls as its CO2 content increases. As the pH of blood falls, its CO2 content increases, so that when partial pressure of CO2 rises, the curve to the right side and the P50 rises. The pH of blood falls as its CO2 content increases. As the CO2 content increases, the pH of the blood falls, so that when PCO2 rises, the curve PCO2 rises, the curve shifts to the right side and P50 rises. So the pH of blood is very important factor, but it is not a direct factor because CO2 is responsible for controlling the pH. The pH of blood falls, decreases acidosis, increase amount of CO2. When it's increase amount of CO2, the curve will shift to the right side and P50 rises. In the tissue, partial pressure of oxygen is between 10 to 40 millimeters of mercury. So in the tissues, the partial pressure of oxygen is 10 to 40 millimeters of mercury and partial pressure of CO2 is high around 45 millimeters of mercury. So an active tissue will have high PCO2, low pH, raised temperature leading to the dissociation of oxygen. Oxygenated blood passing through the inactive cells does not give up oxygen even if its PCO2 is low. But active cells readily gives oxygen as PCO2 is very high. In the tissue, PO2 is between in tissue at the level of tissue, the partial pressure of oxygen is 10 to 40 millimeters of mercury and PCO2 is very high around 45 millimeters of mercury. So an active tissue will have a tissue which is actively, metabolically active. It will gives more and more amount of oxygen. Okay. An active tissue will have high PCO2, low pH, raised temperature. So dissociation of oxygen is, obvious. But oxygenated blood passing through an inactive cell does not give up oxygen, even if its partial pressure of CO2 is low or high. But active cells, the cells which are actively, highly actively metabolically active, okay, they are, producing more and more amount of CO2 gas. Then the oxygen and the dissociation of oxygen from hemoglobin increases, because those tissues, they require more and more amount of oxygen as the tissues are, high at the metabolic rate. So Bohr's effect basically tells us about the effect of CO2, the partial pressure of CO2 is controlling the dissociation of oxygen and hemoglobin. Okay, it was given by Danish physiologist Christian Bohr. The presence of CO2 decreases the affinity of hemoglobin for oxygen and increases the release of oxygen to the tissues. The pH of blood falls, that is CO2 content increases, so that when PCO2 rises, the curve shifts to the right side. This is the Bohr effect versus Haldane effect. Okay, Bohr's effect and Haldane effect, these are the two effects you have to remember. Bohr effect decreases in the oxygen affinity of respiratory pigments such as hemoglobin in response to decreased blood pH resulting from increased CO2 concentrations in the blood. That is obvious. Decrease in the carbon dioxide affinity of hemoglobin in response to increased blood pH resulting from increased oxygen concentrations in the blood. First described by Christian Bohr, first described by John Scott Haldane. Occurs at the metabolizing tissue, occurs in the lungs, describes the release of oxygen, describes the release of CO2, effect under low blood pH, effective under high blood pH, cause the take-up of CO2 at the metabolizing tissues, caused by the uptake of oxygen in the lungs, facilitates the release of oxygen at the metabolizing tissue, facilitates the binding of oxygen to the hemoglobin. When Haldane effect and Bohr effect, they are antagonistic effects. Both are antagonistic effects. Okay, so rest we will discussing the transport of CO2 gas in the next turn. Till then, please take care, stay safe, stay healthy. Bye bye.